Hello my dear students and welcome back to Excellence Batch and I am your Diksha ma'am. So here we have started the chapter Biotechnology Principle and Processes. In the previous lecture we have discussed about the introduction part of biotechnology. What is biotechnology? What are the core techniques in biotechnology and how do we do gene cloning, which is the revolutionized techniques or the fundamentals of your biotech. But how everything is done principally and what are the things that are required, still we have to cover them in detail. So today we'll be talking about what are the tools that we need for biotechnology or for recombinant DNA technology. In the previous class when we were doing gene cloning, I have told you there that during gene cloning we need to cut the DNA. So for that we need some enzyme. We were also joining some DNA. For that we also need some enzyme. So enzyme is one of a tool of biotechnology, right? Just like that I've told you we need some vehicles to transfer the DNA into host organism. So your vector and your host organism is also a tool. Just like before cooking anything, whenever you see any cooking video, what do they say? Well, these are the things that you need to cook the food. You need 250 gram of this, you need 100 gram of this and that. Just like that we also have our recipe, and in that recipe we need certain tools, right? So let's get started. So we have some ingredients here, and these ingredients are known as tools of recombinant DNA technology. Now what are these tools? Let's see. So the first tool are DNA manipulating enzymes. That how can you change the DNA? For that we need certain enzymes. Second thing that we need is a vehicle or vector that will help in the transportation of your DNA, foreign DNA, and the last is host. We'll be discussing vectors in detail later on. Host can be a bacteria, which is E. coli. The most common host that we use is E. coli bacteria, and sometimes we also use yeast, right? So these are the host organism. Host is someone in which you will keep your DNA and then the DNA will divide. Now let's talk about enzyme. We have various type of DNA manipulating enzyme. Like this one, the first one is restriction endonuclease. The most common enzyme, and the discovery of this enzyme was like a boom in the recombinant DNA technology. Without this enzyme, maybe the biotechnology nowadays we are doing, it would be different and it would be little more difficult. Next enzyme that we have is DNA ligase. Another enzyme we have is DNA polymerase, and the last one we have is alkaline phosphatase. So today we'll only be discussing about the enzymes, right? So there are three basic tools. What are these? Enzymes, vector, and host. Enzymes are the enzyme that help in the manipulation of DNA, cut it, join it, replicate it, all these things. So these are four total enzyme important ones: restriction endonuclease, DNA ligase, DNA polymerase, and alkaline phosphatase. These things will, the vectors, which is one of the important topic, we'll be discussing that in later classes, okay? In the later classes. Anyways, let's get started with your restriction enzyme. Restriction enzyme are also known as molecular scissors or knives, right? They are molecular scissors or knives because the name suggests, and it's quite clear from that also. They are going to cut your DNA. They are going to cut your DNA. All right, that's pretty good. But before what they do, let's talk about their discovery. Okay, so first we'll talk about the discovery. How were they discovered? Most of the things in our science, they are discovered accidentally. So this is one of the example. So the discovery took place in 1963. In 1963, there were scientists who were doing a study. And what was the study? The study was, they were seeing that how the E. coli, imagine this is E. coli. E. coli, when it is infected by a bacteriophage, E. coli, when it is infected by a bacteriophage, restrict its growth. Restrict its growth. That means how E. coli is restricting the growth of or multiplication of bacteriophage in it. Because bacteriophage are what? These are viruses. These are viruses that infects bacteria. So what was the experiment they were doing? The experiment they were doing, or what discovery they were meant to make, they were just looking at how E. coli restrict the growth during bacteriophage infection. Bacteriophage infection. This was the experiment they were doing. Now during this experiment, what they realized, what they find out? They find out when the E. coli is getting attacked by the bacteriophage, when bacteriophage is a virus, the E. coli secrete two enzyme. E. coli secrete two enzyme. One enzyme is methylase. One enzyme is methylase and another enzyme was restriction endonuclease. Okay, now why it was doing this and what are the role of these enzymes? So the role of the enzyme is, first enzyme methylase is to add methyl group to the entire DNA of E. coli. So the first enzyme, what it will do, it will add methyl group to the entire DNA of the E. coli. So imagine this green one is the DNA of E. coli. So when E. coli sense that it is going to get attacked by the bacteriophage, E. coli will secrete methylase that will methylate the entire genome of the E. coli. Okay, so it has added methyl group to its own genome. Now it will release the second enzyme, restriction endonuclease. The function of this enzyme is to cut the DNA, to chop the DNA, because it's a molecular scissor, it's a molecular knife. It will cut the entire DNA, whatsoever DNA does not have the methyl group. And which DNA will not have a methyl group? The only DNA which is formed by a virus. So viral DNA at this point is only unmethylated DNA. The DNA of the E. coli itself is all methylated. So the function of the restriction endonuclease, what it did, it cut the unmethylated DNA. Unmethylated DNA. Right? Just for example, the E. coli itself put a shield and now it has triggered a button and from everywhere the bullets are firing. And that bullet will not kill the E. coli's genome. Rather, it will kill the bacteriophage's genome. So it will only cut the unmethylated DNA, and unmethylated DNA is a DNA which do not have a methyl group. And in this case, the bacteriophage DNA does not have a methyl group and hence it will be cut by that, right? So from that we got to know these restriction enzymes are kind of a natural defense mechanisms of the bacteria against viruses, right? So what we got to know by here, first discovery was, yes, there are such kind of enzyme which cut the DNA. Second discovery they came into is that the restriction endonucleases are natural defense mechanism, of what? Bacteria against viruses. Okay, after discovery of this, after 5 years of this discovery, the first restriction endonuclease was isolated and it was identified, it was Hind2. So the first restriction endonuclease was Hind2. was Hind2, and it was extracted from Haemophilus influenza. Which bacteria? Haemophilus influenza. Nowadays, we have over 900 restriction enzymes isolated for over like around 230 strains of bacteria, right? So we have isolated how many restriction enzymes? More than 900 from over 230 strains of bacteria. This is a very important point to note down because the question can be asked. So more than 900 restriction enzymes have been isolated from over 230 strains of bacteria. Okay, so that's about the discovery and which was the first to be discovered. Okay, and isolated. Now, when they discovered the enzyme, they didn't know which enzyme it was. They just knew that this is certain enzyme that can cut the DNA. But the first isolated and discovered was Hind2. Okay. Now, because you must be wondering, ma'am, this is the enzyme, this is E. coli, right? But that bacteria is Haemophilus. How come? Because there they got the discovery. Yes, there are enzymes that can cut the DNA. But from this, they isolated the first one. So first restriction endonuclease to be isolated. Isolated means to take it out, was Hind2. Okay. All right. Now, if I talk about these restriction enzyme, to what class of enzyme do they belong? They belong to a class of enzyme which are nucleases. Nucleases are the enzyme that can cut the nucleic acid. So nucleases are the enzyme that can cut the nucleic acid. What are nucleic acid? DNA and RNA. So they belong to this big class of enzyme known as nucleases, which can cut the nucleic acid. The nucleic acid are your DNA and RNA. Okay. Now the nucleases are of two types. One are exonucleases. The nucleases, this class is further having two categories, and another is endonucleases. Now what's the difference? Exonuclease is a class in which these enzymes will always cut a DNA segment from outside. For example, I have a segment of DNA A, T, 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 T. T, 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 A. Right, this is how the DNA is. Complementary strands, right? Two strands. This is 5' end, this is 3' end, this is 3' end, this is 5' end. Okay, so exonuclease are the enzyme that will always cut at these positions. Outer sides or at the ends of a DNA. They will always cut at the ends of a DNA. They cut at the ends of DNA. Okay, on the other hand, endonucleases. Endo means within. They will always cut within the DNA. How? Like if this is another strand of DNA. Okay, and then we have here 5', 3', 3' and 5' end. They will always cut within like that. They will always cut within the DNA. So they cut within the DNA. To this belongs restriction endonucleases. So restriction endonucleases, they belong to a class of nucleases known as endonucleases. Okay, I hope this is pretty clear. So they will always cut within the DNA. They will never cut at the ends. All right. Now why they are known as restriction endonucleases? Now it's clear they are endonucleases because they are cutting within the DNA. But why are they known as restriction endonucleases? All right, let's unfold this mystery also. So restriction endonucleases got their name restriction from the restriction site. Okay. All right. So how does this restriction endonucleases work? They cannot cut anywhere. It's not like if I have, for example, one restriction endonuclease which is eco R1. Okay, it just cannot cut anywhere in the DNA. It will recognize a particular palindromic sequence. You call it as recognition sequence. For example, in the entire DNA, it will only look for one repetitive sequence, which is known as palindromic sequence. What are palindromic sequences? Which repeat itself. Palindromic sequences. Palindromic sequences are the sequences that repeat itself. Like someone's name is Nitin, N-I-T-I-N. If you read it from reverse, it's still the same. Madam, M-A-D-A-M. If you read it from reverse also, it's still the same, madam. Okay, so these sequences that repeat itself. For example, if I have this restriction enzyme eco R1, it will have this sequence GTTTC, right? Sorry, CTTAAG. So this is a recognition sequence. This is a recognition sequence to which eco R1, which is your restriction endonuclease, can identify. And these are always palindromic, they will repeat itself. So because eco R1 is a restriction enzyme and in the entire DNA, it can only identify this sequence. So these sequences are known as recognition sequences. Recognition sequences, they are always palindromic. That means they will repeat itself. You can see GATTC, GATTC. They will always repeat itself. So what are recognition sequences? Recognition sequences are the sequences to which your restriction endonuclease recognize in a DNA, and they are always palindromic, and they will be around 4 to 8 nucleotides long. 1, 2, 3, 4, 5, 6. It is 6 nucleotide pairs long, right? So recognition sequences are the palindromic sequence of 4 to 8 nucleotide long that is identified by restriction enzyme or restriction endonuclease. RE means restriction endonuclease, right? Okay, next. Alright, so the story doesn't end here. Alright, so if I say this is a recognition sequence of eco R1, this is a recognition sequence of eco R1. So this recognition sequence is a sequence where the eco R1 will stop because eco R1 knows what it is. But now eco R1 also have to perform a function, and that is cutting the DNA, because this is what endonucleases are supposed to do. So restriction endonuclease, which is your eco R1, it will cut at a specific direction at a specific site. You call it as restriction site. So restriction site is a site where it will make a nick or where it will cut. So restriction site are also particular. If it is eco R1, it doesn't mean it will cut here. No. The restriction site is also fixed. The recognition sequence for a restriction enzyme is also fixed. So it will make a cut here. And when it will make a cut, which bonds will be broken? It will always break the bonds in between the nucleotide. And what is the bond in between the nucleotide? This bond. This bond, the red one which I have drawn. This is a phosphodiester bond. Whereas this bond is a hydrogen bond. This bond is a hydrogen bond. So always restriction endonuclease, they cleave the phosphodiester bond. Remember this thing. Restriction endonuclease breaks the phosphodiester bond. Phosphodiester bond between the nucleotides. And this is how both the strands will then get separated. All right, let's see in more detail how the working happens. So you must have seen this in the NCERT also. All right, so this is a piece of DNA we have. This is another piece of DNA that we have. Okay, imagine this is a foreign DNA, this is a vector DNA, the plasmid we have done. If you remember what we have done so far, we had a plasmid, we made a cut in it, and then we had a foreign DNA that we already had cut from the main genome, and then we add it and we make this the recombinant DNA. This is what we have done. So for cutting the vector and for cutting the foreign DNA, we will be needing restriction enzyme. For example, this is your human DNA and we need to cut the sequence of it from it. Similarly, this is a plasmid's DNA. They have not made it complete, they have just shown the little part of DNA. So for cutting both of them, we will be needing restriction enzyme. Another point to note here is that whenever you are cutting vector and the foreign DNA, you will use same restriction enzyme. So mark this line: to cut the vector and foreign DNA means this one and this one, if it needs to get broken down, if you want to cut it, to cut the vector and foreign DNA, same restriction enzyme should be used. Same restriction enzyme should be used. For example, if I'm using EcoR1 to cut the foreign DNA, I should be using EcoR1 to cut the vector DNA also. Why? There is a reason. For example, this is one thing that is happening. We are cutting the vector DNA. Can you all see that? If you can't see that, let me just make it more clear. This is G, A, T, T, C. C, T, T, A, G. So this is a recognition sequence of simply EcoR1. So EcoR1 will be making nicks here and cutting it. When you will cut it, you will get ends. You will get end. How it will get cut? This is how it will get cut. This portion will remain separated. This portion will go away. This is how it will get cut. And here you will see, you will get G, C, T, T, E and A. Now if you want to join any DNA with this DNA, which is a vector DNA, then your foreign DNA should also have the sequences complementary to this. If I want to join anything with this cut piece, I should have A, T and T complementary to this one. Yes, and how can I get the complementary to this one? Only if I will use the same restriction enzyme. So in this case also, we have the same recognition sequence GAATTC, GAATTC, and same way we are making the cut. And how the cut is made? Like this. Okay, like this, and we have got the complementary strands. So now this strand has been formed, which is complementary A, T, T, and here we have C and G, just like G and C here. Now because these strands are complementary, being complementary, they can easily stick on each other here, and then once they got stick, then they will get combined and a recombinant DNA will be formed. What will be formed? A recombinant DNA will be formed. If you can notice here, there is a particular shape of the ends that has been formed. If you have noticed, the shape of the end is something like this. The shapes of the end is something like this. These ends are known as sticky ends. What do you call them as? Sticky or cohesive ends. There are one more ends that are formed, which are known as blunt end or the flush ends. I'll draw that again, right? So these type of ends are sticky end or cohesive end because they are very easy to stick. They are cohesive. They stick nicely to each other. The another type of ends that are formed are blunt ends. How they are formed? Imagine you have a piece of DNA. You have a piece of DNA and then you have the recognition sequence CCC, GGG. This one. This is your vector. And then you have a foreign DNA also. Let me draw it with another color. You have here vector and then you have a foreign DNA. Okay. Then you have foreign DNA. In foreign DNA also, what are we using? In foreign DNA also, we are using here the same CCC, GG, CCC, GG and G. Okay, now the restriction enzyme that we need to use here to cut to make the complementary ends will be SMA1. SMA1 will cut them here. Sorry. SMA1 will cut them here. SMA1 will cut them here. It happens sometimes to the teacher when they are more excited to teach a topic. Okay, so SMA1 will cut them in the middle. When SMA1 will cut them in the middle, then what will happen? What will happen? So when SMA1 will going to cut it, what type of ends will be formed? These type of ends will look something like this, and this piece gone away. Okay, similarly here what will be formed? G, 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 C, 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 like that. Alright, so these ends you can see they are blunt end. If you'll see the design, it's blunt like this. So you call these ends as blunt or flush end. They are more difficult to stick. If you want to join this foreign DNA and vector DNA, you will need to have more energy to join them as comparison to the sticky end, because they stick easily. Why do they stick easily? Why not the blunt ends can stick easily? What happened? The sticky ends, let me draw it here, G, T, like these were your sticky ends formed. Okay, how the sticky ends were formed? Like this. This was one end, imagine, of a vector, and this was under another end of the foreign DNA. Right? This was like that. So whenever they will come close to each other, they don't need to make a phosphodiester bond first in between these two. They will first form hydrogen bond. And once the hydrogen bonds are formed in between the complementary strands, it will be very easy to form phosphodiester bond between them, because phosphodiester bond is a more high energy bond and it is difficult them to form. Here, if you want to join them, straight away you have to go for phosphodiester bond. So that's why sticky ends are more easy to stick, because they being complementary to each other, they can easily form hydrogen bonds first, and then they can form phosphodiester bonds later. So note this line with me. Sticky ends are more easier to stick because complementary strands forms hydrogen bonds first. Okay, that's why they're more easier to stick. So two types of ends can be formed. That was about how these work. Let's talk about their nomenclature. Why do they have weird names? Right? Why do they have weird name? So if you have noticed, the name of one such restriction enzyme was EcoR1. So there is a rule. What's the rule? The first will always be the name of a genus. Like in case of here, we have Escherichia, the name of a genus. Second will be the name of the species, like coli. So that's the name of a species. And then this letter will be for the strain. And the last, which is 1, 2 or 3, will be according to order of discovery. Will be according to order of discovery. And this is how you have given names to all the restriction enzyme. First the genus, species, strain, and then the order of discovery. Alright, let's see how restriction enzyme helps in gene cloning. The same we have done in the previous class. First of all, always have a foreign DNA, cut it with the restriction enzyme, the same restriction enzyme you are using to cut the vector. After it has been cut, then you're going to join them. When two pieces are joined, they become a recombinant DNA, which is sent into the host organism, which will which is known as transformation, and which will then multiply and form new pieces of your foreign DNA along with its multiplication. So this is how restriction enzymes plays a very, very important role here. Now comes the role of the second enzyme, which is DNA ligase. Let's talk about other enzymes also. The next enzyme that we have is a DNA ligase. Ligase. I say ligase in the G in the ligase tells about itself. That means they are molecular glue. They are molecular glue. So what does glue do? Scissors were restriction endonuclease and glues are ligases. So what does glue do? Glue stick two things together. So what are they doing? They join the pieces of DNA. Or better, I would say they will join vector and foreign DNA. So if I want to join vector and foreign DNA, for example, CCC, GGG, this is the one piece of DNA. This is the another piece of DNA, GGG, CCC. Now I want to join them. So I will join them with the help of formation of these bonds, and the name of the bonds is phosphodiester bond. So simple funda. These bonds, they are cut down by restriction endonucleases, but they are joined by DNA ligases. So the DNA ligase, a particular DNA ligase that we use here is T4 ligase. Why T4? T4 is the name of the virus from which it was discovered and isolated. It was discovered and isolated. Okay, so this is what we are using to join the DNA. That is DNA glue. So the second step which we do in this DNA gene cloning is joining these two pieces of DNA, and that happen with the help of enzyme DNA ligase. And in this case, the most commonly used is T4 ligase. Without this, again, recombinant DNA technology is incomplete. Now, once the DNA ligase has done its thing, now it will go to the host. In host, the replication occurs. For replication, we need to use another enzyme, and that is DNA polymerase. So DNA polymerase 1 is used for DNA replication. So we want that our piece of DNA should divide in the host. So we will also provide them DNA polymerase 1 so that the multiplication should keep on going. Another piece of DNA enzyme that we use is alkaline phosphatase. Now this is something which you have never heard of. This is the enzyme that basically cut phosphate at the 5' end. You all know at 5' end of the DNA we have phosphate group free, and at the 3' end of DNA we have hydroxyl group which is free. Yes, you all know that, na? No, you don't know. If I have this 5' end, like we have A, T, T sequence. This is 3' end. This is 3' end, T, 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 A sequence. This is how DNA is. So why does 5' is known as 5'? Because at 5' end, your this nucleotide, it has phosphate group free, and here this one has OH group free. This has OH group free, this has phosphate group free. Okay, now we always know whenever a phosphodiester bond is formed, phosphate and OH, they join, they form bond. This is our phosphodiester bond. To make a phosphodiester bond, you just need a phosphate group and OH group. And here, when you cut the piece of DNA, whether it's your foreign DNA or if it is a vector DNA, they have these phosphate ends and OH ends. Now there is a chances that this piece of DNA will join and become circular. To make a phosphodiester bond, you need phosphate and OH, and we have phosphate OH. They may join and coil and make a circular ends, right? And they make a circle. So to avoid this, alkaline phosphatase cleaves phosphate at 5' end so that the DNA should not circularize. So what does this enzyme do? It cleaves phosphate group at 5' end to prevent the ligation of ends. Ligation of ends. So that's why I need alkaline phosphatase also. So that was all about the enzyme. Let's move further and talk about some questions. So the first question is, okay, the linking of antibiotic resistance gene with the plasmid vector become possible with first option exonucleases, second option DNA ligase, third option endonucleases, fourth DNA polymerase. So first of all, understand what the question is saying. It's a previous year question in 2008 that has been asked. See, all questions that we'll be discussing today, they all are previous year. All right, so that by that you will get to know, or you'll get an idea that how much important this topic is, right? So the question is asking, whenever you are linking antibiotic resistant gene, which gene is they talking about? Antibiotic resistant gene with plasmid. What is it? With plasmid. If you want to join these two genes, what you will be needing? You needing molecular glue because you're joining things. So for that we need DNA ligase enzyme, and which is the most common DNA ligase used? That is T4 ligase. Alright, so answer to this question is second. Next question. Restriction endonuclease. Cut the DNA molecule randomly. Second option, cut the DNA molecule at specific site. Restrict the synthesis of DNA inside the nucleus. Synthesizes DNA. Okay. First one says that this enzyme, which is restriction endonuclease, it can cut DNA at any site. This is absolutely wrong. It only cut at a restriction site of a recognition sequence. It just cannot cut anywhere. Cut the DNA at specific site. Very true. It does not restrict the growth, it's just for cutting, and it does not synthesize DNA. The function of synthesize or DNA replication is of DNA polymerase. And the most common DNA polymerase used is DNA polymerase 1 in case of recombinant DNA technology. So, the answer to this question will be 2. Next, given below is a sample of a portion of DNA strand giving the base sequence of the opposite strands on the opposite strands. What is so special shown in it? Okay, so they have given you a strand of DNA. GAA, TTC, CTT, AG. So those who have done biomolecule very nicely, if this question is there in your biology section of NEET, you will straight away get to know this is from biotechnology. Okay. Now what is special in this? Palindromic sequence of base pair, replication completed, deletion mutation, start codon at the 5' end. Okay, so first of all, this is not a start codon, and this does not tell us anything about mutation. To know about mutation, you need to know about 2 strand after and before thing. Okay, and replication completed, we also don't know whether the replication is done, not if the strand has been a cut portion only. But yes, it shows a palindromic sequence. What is a palindromic sequence? The sequence that repeat itself. GAATTC, GAATTC. If you can see here, it repeat itself. Okay, so answer to this question will be 1. Next, the DNA molecule to which the gene of interest is integrated for cloning is called carrier transformer vector template. So foreign DNA or gene of interest is simply carried by a vehicle named as vector. So answer is 3. This is the simplest form of question from gene cloning, right? Okay, though vector will be discussing in next class also in more detail. The cutting of DNA at specific location become possible with the discovery of ligase, restriction enzyme, probes and selectable marker. Earlier the question was on the linking of DNA, that the DNA has been linked with the help of molecular glue. Now it is on the cutting of DNA. So for cutting we need molecular scissor, that is restriction enzyme. So if you have noticed, all our previous year question and all are straightforward question, nothing complicated, right? Next, Which of the following is a restriction endonuclease? RNase, HIN2, protease, DNase 1. Very simple, HIN2. HIN2 is restriction endonuclease. You can, if you even don't know, but you know their nomenclature is done on the basis of their names, you can simply mark it. Right? Next. Following statements describe the characteristics of the enzyme restriction endonuclease. Identify the incorrect statement. Okay, so first of all, see what first statement say and what second says. First says that there are certain statement on the restriction endonuclease. Okay, so our mind is prepared for biotech. Now what you have to find in them? You have to find the wrong statement, the incorrect one. Let's get started. First one, the enzyme cut DNA molecule at identified position within the DNA. Second, the enzyme bind DNA at specific site and cut only one of the two strands. Third, the enzyme cut the sugar phosphate backbone at specific site on each strand. And fourth, the enzyme recognizes a specific palindromic nucleotide sequence in the DNA. Okay, let's see. The first one say the enzyme, the restriction endonuclease, cut DNA at identified position, the position it can identify, which are recognition sequences. This is true. Enzyme bind DNA at specific site, absolutely true. And cut only one strand? No, both the strand. It will always cut both the strand. If you remember, we have done GAA-TTC. Yes, so this is a sequence of DNA which is recognized by EcoR1 and it will always cut here and here. So that means the restriction site is on both these strands. It will be cutting the phosphodiester bond of both these strands, not the single strand. Okay, that's why the complete piece of DNA will get out. Right, so that's the incorrect. Let's see, are these correct or not? The enzyme cut the sugar phosphate backbone at specific site on each strand. This is also true. The site is also specific. It is not that EcoR1 is cutting here, somewhere here, somewhere here. No. Every time if EcoR1 is cutting, it will cut here only, in between G and A. Okay. The enzyme recognizes specific palindromic nucleotide sequence in the DNA. So the recognition sequence are always palindromic, which are identified by the enzyme. That means they will repeat itself, just like GAA, TTC, GAA, TTC. It repeats itself. Fine. So answer to this question straight away will be second. This is incorrect statement. Next. Match the following enzyme. It's a 2019 question. The recent, not the recent one, but yeah, than the others. Match the following enzyme with their function. Select the correct option from the following. In column A, we have restriction endonuclease, restriction exonuclease, DNA ligase and Taq polymerase. Here we have join the DNA fragment, extend primers on genomic DNA template, cut DNA at specific position, removes nucleotides from the ends of DNA. Alright, so endonuclease will always cut the DNA within, not at the ends, and always at a specific position. So for A we have 3. Exonuclease will always remove the nucleotides from the ends of DNA. That's why it is known as exo. Exo means outer, endo means within. DNA ligase is the one which will always join the fragments, and Taq polymerase will be doing in the PCR, polymerase chain reaction. Okay, so this is the one which will extend the primers. It's like a DNA polymerase. So for this we have 2. So A3, B4, C1, D2. Answer to this question is 3. Simple. Next. An enzyme catalyzing the removal of nucleotide from ends of the DNA is protease, DNA ligase, endonuclease, exonuclease. Very simple. It is removing the nucleotide from ends. And which one removed from the ends? Exonuclease. That is 4. Endonuclease always will remove the DNA within. So you know this question is also kind of a confusing. There are some students who conclude the answer just by reading the first half of the sentence. Like an enzyme catalyzing the removal of nucleotide, but they forget to put emphasis on from ends. So otherwise in first go, if you read it partly, then you will be giving the answer 3. but read the answer, read the question statements very carefully. Okay, so answer is 4. Next. That's it. So this was all about restriction enzyme. Trust me, from NEET point of view, this topic is really very important. You will definitely get questions in NEET from this topic somehow, whether it is a statement based question or whether it's a direct question or something like that. So what you have to do is, the homework is to read the NCERT, solve the question from module, and be ready for the next class. In next class we'll be discussing a little more important topic, that is vectors, and it has a lot of conceptual things in it. So refresh your mind before attending my next class. Till then, bye bye, take care, work hard.